Hey, it's Miss Segovia again. So far you should have watched videos on carbohydrates, lipids, and proteins, but we're going to tie all those together with a really brief introduction to nucleic acids. Nucleic acids are the information storage that helps make all our other macromolecules possible. The function of these is genetic material, storing the information into the genes, which is a blueprint for building proteins. Our whole central dogma of biology is that DNA creates RNA, which creates our proteins. And so it's the big process we will be covering far more in depth in the upcoming days than we are in this little video snippet. That information can also be transferred in the blueprint for new cells, and so our little duplication of our cells here for our next generation. And you guys all know everybody's favorite topic in biology is reproduction, making more cells. And so we will be studying lots and lots of information on DNA. So again, just super basics in here. DNA can, nucleic acids can be made into RNA, which is ribonucleic acid in a single helix structure. And we can also have DNA, which is our deoxyribonucleic acid, and our double helix structure that you guys are super familiar with by now. The monomer of nucleic acid is the nucleotide. We love DNA made of nucleotides. Sugar, phosphate, and a base bonded down each side. So the three parts here, we have our nitrogen base, our A, T, C, G, or U, adenine, thymine, cytosine, guanine, or uracil. We have our pentose sugar, which is a 5-carbon sugar. That's why it's called a pentose. In RNA, it's ribose is their sugar. In DNA, it's deoxyribose. So what does RNA have that DNA doesn't? Oxygen. So really simple little molecule structure there, right? Super simple. Our phosphate group then is our PO4 group. And this is a really important one because it forms the linkage from each one to the next. And when we get a little bit more in depth, you'll see how it actually helps in the bonding of our DNA and RNA molecules. So we call these guys charged because if you look at our phosphate group, you'll see these little negatives right here. And so DNA actually has a negative charge. And that's going to be a big deal when we start talking about gel electrophoresis and the movement of DNA through a gel so we can analyze it further. We have to have that negative charge in order to make it happen. So DNA will actually form by the pairing of our nucleotides. They have hydrogen bonds, 2 between the A's and T's and 3 between the G's and Z's, our purine-pyrimidine bonds. And we will form those down the center. We want to match them up so that we can preserve the order that they're in. Because as we talked about in proteins, the order is super important so we don't end up with mutations that can have really awful consequences. So today, again, we're not going in depth on DNA at this time, but I do want you to have a little DNA molecule in your notebook. You'll notice on this one you are not putting those neat little water droplets because DNA does not form through dehydration synthesis. It's kind of our little oddball of our macromolecules. So right now, just go ahead and on the top of page 11, cut out 2 strands of DNA and bond them together at the center and glue that up at the top. And give it way up at the top because you're going to need plenty of space for our little slider that we'll show you how to do here in a bit. Thank you for your time.